Thank you very much for the kind invitation. My name is Olga Vitek. I'm a faculty in the Curie College of Computer Sciences at Northeastern University. I am a statistician by training. My group is interested in all things quantitative mass spectrometry and proteomics. And in particular, we are developers and maintainers of MS-Stats and MS-Stats TMT, which is an open source software for design and analysis of quantitative mass spectrometry-based proteomic experiments. And today I am very excited to share with you what we do specifically in the MS-Stats and MS-Stats TMT space. And the hope is that it will be useful for many of you. So I will start right away. So MS-Stats is a open source software and a series of methods to support quantitative proteomic experiments. It is distributed through R in Bioconductor. And this effort started in my lab 10 plus years ago. And it started with a very simple question. Which proteins change in abundance between conditions, such as healthy and disease? And it sounds simple enough, but very quickly we realized that this is actually a fairly complicated question for many reasons. And the first reason is complex design. So in reality, we rarely are interested in just two conditions, such as healthy and disease, but often we want to incorporate multiple related conditions, such as multiple related treatments, or maybe different subtypes of disease, and so on, into our study. And these multiple conditions, they affect the statistical structure, stochastic structure of the data. And of course, there are other types of designs which are very popular and very effective, such as paired designs and time course. So for example, paired design would be where you have a healthy and tumor biopsy on the same person, and time course when you collect repeated measures on the same person in time. And these type of designs, they also require a different type of statistical analysis. And then very quickly, we realized that there are many other related questions which we can also attempt to answer, such as post-translational modifications. So which PTMs change in abundance or in amount between conditions, but distinguish that from the overall change in protein abundance. So how can we look at changes in PTM and distinguish them from changes in overall protein amounts? And then also structural biology, limited proteolysis also can be used to, can be analyzed using similar type of methods. And also byproduct of these methods is, or questions, is to quantify the abundance of a protein in a single sample on some scale, which is relative but is comparable between the runs. So we have multiple spectral features. Can we summarize them somehow so that we can compare this feature, these protein abundances between runs for the purpose, for example, of classification or clustering, or just for the purpose of visualization? So these are related questions, and at this point, MS-Stats can be used to answer all of them. And of course, the design is not the only aspect of these experiments. Another important aspect is how the samples are prepared and how the data are acquired. And in particular, we can distinguish data-dependent acquisition label-free versus labeled, for example, with TMT, data-independent acquisition, parallel reaction monitoring, selected reaction monitoring, etc. So MS-Stats can work with all of these acquisition types. Now, once the data are acquired, the first step in the analysis is actually to identify the peptide ions which generated the peaks and map them to protein sequences, and also map them between the runs. And these are fairly complex procedures, and in MS-Stats we do not do that. Instead, we work with other scientists and other developers of tools which do this work. And we are trying to be as equal opportunity collaborators as possible. And we collaborate with the majority of open source and also commercial tools that are developed for this purpose, such as Proteome Discover, of course, and Spectronaut as far as commercial tools, and also open source and open access tools such as Skyline, MaxQuant, FragPipe, Diane, etc. And when we take the output of these tools as input to our methods, well, this data need to be prepared in the right format. And we developed many converters that do that. And they don't just prepare and massage the data, but they also do additional data analysis and preparation steps, which consider the quantitative information between the runs. So, for example, we can filter out some features which are redundant. For example, if you use decoy sequences and these decoy sequences somehow made it into the data set, we can eliminate that. We can eliminate features which are have too many missing values, or we have options whether we keep for the purpose of protein level quantification if we keep just the unique peptides, or if we also allow shared peptides. So this is all really important steps, and in fact, in our experience, this affects the quality and the type of statistical conclusions downstream quite a bit. Of course, the core of MS stats is the statistical analysis, which happens once the data are read into R and into MS stats. And there are two aspects. Well, one aspect is we need to model the stochastic structure of the data appropriately in a way that accounts for the experimental design, accounts for outliers, accounts for missing values, accounts for the multiplicity of proteins, multiplicity of features, potentially for the fact that different features have different extent of variability, etc. And so we have a variety of models which are available, and MS Stats recognizes the experimental design and the acquisition automatically and chooses and fits the right model so that the tool can be used by users with limited or nearly no statistical expertise. And then the other aspect of that is numeric stability and scalability, because of course we feed these models many times over for different proteins, and things have to be numerically stable. For example, if a particular protein has a pattern of missing values such that the model cannot be fit, some other model will be fit that is appropriate. And just numerically, things have to be well calibrated and well behaving. So we pay a lot of attention to that. And then, of course, once we can do all of that, a very important aspect is how can we learn from this data to plan future experiments. And we have a series of functionalities for that purpose. In particular, the most frequent question that we are asked is how many replicates do we need? So for that, we need to understand how much variability we have in the system. And for that, we can use the current data set as a pilot study for something else and infer sample size. And also we have system suitability. So this is methods from process control where we can monitor standards that are either interleaved between the samples or spiked into the samples and detect deviations from optimal performance. And before that, even method validation, calibration of an assays in terms of limits of detection, limits of quantification. So we have methods for that as well. So this is the big picture in terms of what MS Stats can do. And so, as I mentioned, MS Stats is methods. And at this point, it's not an R package. It's a family of R packages, which has as input all these different types of tools, output of all these tools. And we have the one package, which is MS Stats Convert, which takes care specifically of the conversion. MS Stats is the base MS Stats package, which is designed primarily for label-free experiments. MS Stats TMT is built on top of that, and this is what we'll focus on today. MS Stats PTM can work with both label-free and TMT labeled methods, and also MS Stats LIP. And then the output of MS Stats is compatible with Massive Quant. So this is an extension to the Massive repository where you can store the results of your quantitative analysis and also all the analysis intermediate steps and scripts and parameter files, etc., to make sure that your entire quantitative analysis workflow is reproducible. Now, most frequently, MS Stats is used as an R tool through the command line. However, increasingly, we have users who do not have this ability to use R programming, And so now MStats is accessible through two types of graphical user interface. One is a Galaxy project. Galaxy is a general framework which enables the analysis of genomic experiments and proteomic experiments, and you can build workflows which are specific to your project, and MStats workflow is one of them. And we also have implemented a dedicated graphical user interface for MStats called MStats Shiny. Shiny is an R framework to build graphical user interfaces, and most of the functionalities now are also available through MStats. And before I continue, I would like to make sure that I can acknowledge everybody who worked on that. Mina Choi was at the origin of the MStats project and made really important contributions, especially to the core MStats package. Ting is the lead author of MStats TMT. Devon has developed MStats PTM, MStats Lib, and also MStats Shiny. Mateusz is a software engineer who did a lot of backend work to make sure that numerically all the methods are scalable and stable. And Melanie has led the integration with the Galaxy project. Okay, so the plan for today, I will spend most of the time talking about MStats TMT, and then I will show you also how in the context of TMT labeling, we can also extend these methods to post-translational modifications, and also how this can be accessible through the graphical user interface. So very generally, the way we think about MStats statistical type of workflows is like that. Before we collect data, we need to make sure that we understand the nature of the experimental design and also the nature of the question we want to ask from the data. And in the context of TMT labeling, in addition to many different conditions, we also need to think about how we can allocate samples to mixtures. So in terms of complex design, let me just give you a few examples. So this is a experiment which we had a collaboration a long time ago, where we have two types of breast cancer cell lines, MCF7 and HS57AT. Each of these cell lines had two types of oxygen exposure, normoxia and hypoxia. Each oxygen exposure was considered for two different time points, 6 hours and 24 hours. Each of these exposures had two replicate cell cultures. And then for each cell culture, there were also multiple mass spectrometry injections. So now we have a fairly complex experiment where, well, we may want to compare, let's say, 6 to 24 hours of hypoxia for this cell line, but maybe we also want to ask a more complicated question, such as, is the change in oxygen exposure for the invasive cell line similar to the change in oxygen exposure for the low invasive cell line, and things like that. So we can ask composite questions, and MStats can specify the experimental design corresponding to this experiment, and it can ask this complex composite question. And it is set up in a way that you do not really need a whole lot of statistical expertise. Here's another example. This is a time course experiment. So this was childhood cancer study where we have 29 healthy subjects profiled at only one time point. And then these are the disease patients which were profiled first at diagnosis, then at different time points during chemotherapy, before surgery, after surgery. And so it's a combination of a group comparison design, healthy versus disease, but also time course repeated measures design for the disease patients. And we can ask questions such as, well, which proteins are differentially abundant between healthy and disease at diagnosis? But we can also ask questions, which proteins during the course of therapy became more similar in abundance to the controls than before. So again, this is a combination of variability in repeated measures versus group comparison, complex comparison. MStats can do that. And then in terms of allocation of samples to mixtures, we will spend quite some time talking about this in a minute. So once the data are collected, the input for us is list of identified peptide spectrum matches with quantified reporter ions. And we have converters from Proteome Discover. Proteome Discover even has a scripting node for MS Stats, and of course, many others like MaxQuant Spectronaut, FragPipe, etc. And as far as converter work, as I mentioned, we can filter out shared peptides. It's an option, so you can control that. PSM with many missing reporter ions. For peptide ions with multiple PSM, we keep best PSM with respect to intensity, missing values, interference score if reported, etc. I forgot to mention, if the experiment contains fractions, we can also combine fractions, and we do it in a fairly simple manner. If the same peptide ion is present in multiple functions, we only keep one copy, which is most in the function where it is most abundant. So the input to MS-DAS is the output, for example, of Proteome Discover. So here we have one line is a feature intensity, and then feature intensity is annotated with things such as protein name, peptide sequence, charge, from which mixture it came, from which run, in case we have multiple multiple injections of the same mixture, and so on. So this would be the workflow of the converter, and again, it would include various types of filtering. So now the important thing for us is, how do we actually work with this data structure on the statistical modeling side? So for this, I think it's helpful to contrast what happens in TMT versus what happens in label-free experiments. So this is how we think in MS Stats about a experiment, a label-free experiment, just one protein. This is a array. The columns in this array, this would be mass spectrometry runs. And in case of a label-free experiment, maybe we have multiple technical replicate runs from the same subject. So all these columns, they are grouped by a same subject. Now, multiple subjects, they are grouped because they come from the same condition. And then we have another condition where we have potentially other subjects. This is group comparison design, or same subjects if this condition is a different time point. And so the pink part of this data structure is actually the part of the experimental design, which is of interest to us. That's how we set up the experiment. And then the rows, these are spectral features. And in each cell, the little letter Y indicates the intensity of this feature. And as we can see, sometimes these feature intensities can be missing. And so in MS Stats, when we work with label-free experiments, what we do, we split it into the analysis into two steps. First, we ignore the pink part of the table. We only summarize the yellow part into one number per protein per run. And then we focus on the pink part to model the experimental design appropriately. But in TMT, it's more complicated. And this is why it was required for us to create a separate package, because we couldn't simply extend the label-free version of MS Stats for TMT. So here we have a different statistical structure of the data. If you look at the top grouping, the blue part is the mixture. So the mixture groups, and mixture, first of all, can have multiple mass spectrometry technical runs, right? So multiple injections of the same mixture. And then within each run, we have multiple reporter ion channels, which can have multiple conditions. And then within each condition, we can have multiple biological replicates. So this is a really important difference, because here, the grouping of the runs is essentially governed by the biology, right, by the experimental design. Whereas in TMT, the major grouping is governed by the technological aspect, which is the pool of the samples into one mixture. And the biological aspects of the experiment, which are of interest to us, so conditions and biological replicates, they're kind of buried within this hierarchy. So this is one distinction. And then another distinction is, well, now we have the rows in this data structure. They are features. And unlike in label-free experiments, where we either map MS1 peaks or fragments between the runs, or we propagate the intensities somehow. Here we have DDA MS2 spectra, and we have much less ability to essentially propagate the same feature across multiple runs. So here, the small symbol X means that we observe for this feature, and the feature is a peptide ion, right, a PSM. So we observe this PSM across multiple technical replicates and multiple mixtures. Occasionally, we have one missing reporter ion channel, but it doesn't happen very often. More often, however, we have in one mixture, we observe the PSM with the majority of the reporter ions, and then in the next mixture, we do not observe this same PSM. And so now we have, if we really think of our features as all possible PSM which were observed for this protein in at least one run, then we have a data structure which has a lot more missing values because we don't have the same PSMs in different mixtures. And this is somewhat of a problem because, of course, different peptide sequences ionize differently, and they were acquired like from at different points in the chromatographic peak. And so these intensities in the feature in run one are not necessarily directly comparable with intensities of a different feature in run two. And so our goal is to integrate this information across multiple mixtures and across multiple runs so that we can compare the intensities of multiple replicates, multiple conditions in all the experiment. So to be more specific, so this is one example. So this is a controlled mixture. This is how MS-Stats typically visualizes that. So I see that the quality of the image doesn't come out really well, but let's try anyway. So here, each panel is a mass spectrometry run, and the dashed lines in each panel separate the conditions. So the conditions are annotated with the abundance of a particular protein. And within each condition, we have two replicate reporter ion panels. So here we have 15 runs, total 15 different runs for this experiment. And we have 5 conditions within each run. So now the y-axis here is the log intensity of the reporter ions, of the peptide ion sequences of PSMs for a particular protein. This is the protein name. And so this is the label of different peptides. And so usually everything we do after log transform because statistical modeling is facilitated on the log scale. And so what you see is that we don't have the same colors essentially in every run, right? So for example, this pink feature is somehow low intensity and variable in the first run. It is absent from run 2. It is absent from run 7, etc. Run 2 has only 2 PSM for this protein, green and yellow, but then some other runs, for example, run 15, have a lot more PSMs. And so our goal now is to integrate this information, understanding that the main grouping here is with respect to the run and mixture, and make sure that the summary intensity of the protein over all the features are comparable not only across different reporter ions of the same mixture, but also across the mixtures. So that's the idea. So let me now tell you about how we actually do that in terms of statistical and analysis workflow. And essentially, to make things comparable, we rely on normalization and statistical modeling. So the first step is spectral level normalization. Now, here again, each panel is a run or a mixture. Each box here is a channel. And the y-axis is the log intensity of everything in this run. All PSMs, all proteins, all of it. And assuming that in this experiment, only a small proportion of proteins is actually differentially abundant, we would expect that the median intensity in all of the channels will be roughly the same. If the experiment is such that some conditions have all the proteins regulated, this assumption is not appropriate. But in many experiments, we can assume that the majority of the proteins are not regulated. And so then the differences in the median intensity there rather due to labeling efficiency or some kind of technological artifacts than to the biology. And so the first step is to equalize the medians of all the channels. So we call it global spectral level normalization. It is global because it does the same thing for all the proteins at once. So then the second step is summarizing our features into proteins. So here again, each panel is a run. The gray lines are PSM, so all the ions, but now for one protein. So this figure corresponds to only one protein. And what we will do, we'll summarize all these features into one number per channel, per protein. And we do it the same way as we do it for MS stats using a robust summarization. You can think of this as a glorified median. It's called Tukey median polish. And so now we have one number per protein per run. So some details on how we do this. Maybe just before that, let me make sure we notice that these summaries are still not comparable between the runs, right? Because these PSMs here may be in run 1 may be very different from PSMs in run 2. And so now within the run, different channels are comparable, but between the runs, they're not comparable. So just a small detail on the summary. So we do this type of robust summarization, but we also account for potential missing values. In TMT, a single channel missing doesn't happen very often, but it happens sometimes. And so what MS Stats is doing, it is essentially using this model to also do the imputation of missing values. Here is one example. Let's imagine that we have this type of situation where we have one channel blue and one channel red, and we have three PSM. This is just one protein, so we have three PSMs for the protein, 3 measurements in the blue channel and 3 measurements in the red channel. But now let's imagine that the lowest blue measurement is actually below the limit of quantification and it's missing. So at the very left, if we just ignore it and keep it missing, we see that the summary of 2 blue points will be slightly higher than it should be. So we will underestimate the effect of regulation. Then on the other hand, if we impute with something very small, like 0, then the summary of the 3 blue dots will be lower than it should be. And we would overestimate the effect of regulation. And in MS Stats, what we do, we would make the assumption that all the PSMs of the proteins are supposed to be parallel because they come from the same protein. And as long as we have another feature for the same protein in the same run, and as long as we have this same feature in some other channel, we can essentially draw the straight line and we will impute the missing value based on this parallel pattern. In label-free experiments, that's actually quite impactful. In TMT, occasionally it makes a difference, but not as big a difference as in label-free experiments. Okay, so now we have a summary of one number per channel per run or per mixture, plex. However, things are still not comparable between the runs. And so our goal now is to make sure that the mixtures are comparable between the runs. And so for this, we need to do protein level normalization. And so now this is what we call local normalization because it is performed separately for each protein. And it specifically is designed to make sure that we can compare intensities between runs despite the fact that they came from different PSM. And for that, what we very much recommend is to have at least one channel in each plex, in each mixture, which is a reference channel, meaning it is the same biological material present in every mixture, which does not have any meaningful biological variation. It can be some pooled sample, it can be some just consistent reference sample, but we know it doesn't have a biological variation. So here's a cartoon where we have 2 reference samples per run. And we see that like the red line is roughly the median of those. So we see that in run 1, the reference is slightly below the red line. Run 2 reference is roughly on the red line. Run 3 reference is roughly above the red line. So what we will do, we'll equalize all of this reference channels, and then we will apply the same shifts to every other channel in the run to compensate for the fact that we have different PSMs. This cartoon has 2 reference channels. We looked into that. it helps to have two reference channels, but then if you can free one channel for an extra biological replicate, it's better. So if you have extra space, good to have two references, but if not, one reference is enough, but we need to have at least one. There are some methods that propose to do this normalization without the reference channel, but they make very strong assumptions. And the reference channel allows us essentially to avoid these assumptions when they're not necessary. And then the last part is modeling and inference. And in this case, we need to make sure that we account for the fact that we still have multiple plexes. And this is the part that I would like to now kind of talk about. And the rest of the topics I will mention very briefly. So let's go back to the fact that we have many ways of allocating samples to mixtures. And this is really a key challenge in TMT experiments, which have more replicates or more conditions that can fit into one plex. Here is one example from a CPTAC study, fairly recent, where scientists had 23 mixtures, and it was a paired design because each patient had both tumor and adjacent healthy biopsy. And so they allocated each patient within the same plex. Within plex, we can have 4 patients, and then they had many more replicates across mixtures. And importantly, there was a reference channel. Another example, which is kind of showing less balanced design in the sense that here we had 3 conditions, but they didn't have the same number of replicates within each mixture. Again, notice that we have a reference channel. And, like sometimes we have experiments that look like that, where we have some kind of really, non-systematic allocation of samples of conditions. And there's a reference channel, but we have different channels and different samples. Some mixtures have multiple references, others have single reference. The point is that all of this creates additional complexity, but MS stats will know how to handle that. So what is the challenge, and what are the things that we need to be aware of when we analyze this experiment? So I won't do much mathematics, but I just hope to give you intuition. Let's say we have a group comparison design. So we have 2 conditions, and we have different people, different patients in each condition, and they're all allocated to the first mixture, to the first plex. And then we have more replicates, so we have more replicates from each condition in the second plex. I would like to argue that the comparisons within one plex are more precise than comparisons of these subjects to this subject, so comparisons across plexes. So a good way to analyze this data would be first compare conditions within mixture 1, compare conditions within mixture 2, and then combine the comparisons, because within mixture comparisons are more precise. Well, then we have another option. What about time course experiment? So what we can do, we can also have in one mixture, have multiple time course, but now we have space only for one replicate, for one person with multiple times in one mixture, and, well, actually 2 replicates and a second person across multiple times. And then we have more replicates, so we have a second mixture. So now I would like to argue that there are 2 things that are going on. First is the comparison of times within the same person is more precise than comparison of times between people, because each person serves as their own control. And so within person comparison is the most effective way to detect differences. And this is why time course and repeated measures designs are very good designs. But then between person comparison is more noisy, but between person and between mixture comparison is even more noisy. And so now, in terms of statistics, we need to first look for comparisons within a person across time, then aggregate the comparisons into one mixture and aggregate the comparisons in the other mixture, and then combine the aggregated values. And of course, the real life experiment kind of often look like that, where we have multiple conditions, multiple time points, and multiple mixtures. And then now we have between time within person comparison is most precise, between conditions within mixture comparison is more precise, and between mixture comparisons are the least precise ones. And so we need to make this kind of multiple levels of aggregation to leverage the precision which is available to us due to this design. So how do we actually do that? Well, so most of the time, the statistical methods all function in terms of the signal to noise ratio. So what is the signal and what is the noise? And so when I talk about precision of the comparisons, it is really about how do we quantify the uncertainty, how quantify the noise. And so the methods in MS stats work essentially in terms of decomposing the total variation into variation between subjects and within subjects, and between subjects across conditions and within conditions. The important point I want to make here is that in this particular experiment, comparing times within subjects is more precise than comparing conditions. So we need to have different noise, different denominator. And so these models, when we compare conditions, that will determine what is the denominator for this signal to noise ratio and what is the amount of information available in this experiment. And then if we want instead of compare times, we will have a different part of the model which is used as a noise structure, and it would have different amount of information dedicated to that comparison. And again, MS stats can do that. And just in terms of comparisons, so some other methods that you may have heard of, such as lemma, they are not able to separate this variability in so many different levels. And as the result, they cannot distinguish the amount of information available to compare conditions from the amount of information available to compare times given multiplexes. And so here are some examples. So this is a protein where nothing really happens between times and a very small change between conditions. So lemma will think that we have exactly the same amount of information for both, but MS stats will be able to kind of show that comparing conditions, which we have much less information than comparing times. And so this makes a big difference in practice. Okay, so now I just have a few minutes to show you how we can extend this type of concepts to do something potentially even more complicated. But turns out that because the methods in MS stats and MS stats TMT are very general and broad, these extensions are actually not complicated at all. So it looks like it's more complicated, but actually it is not. And so post-translational modifications, this is really one such example. So now we have two data structures. Remember before we had one data structure for one protein, now we have two data structures for the same protein, where on one hand we have features with a modification site, which are actually modified, and then features which do not cover the modification site, and they do not have the modifications. And so each of these data structures is the same as before. So this is an example of label-free, but we can imagine the same thing for TMT. So essentially for the same protein with double data structure, this is all our features which have the modification, and we will use them to characterize changes in PTM and stoichiometry between conditions. And these are the features that do not have modifications, and we will use them to characterize the overall change in protein abundance. By the way, where do these features come from? Typically, it is really useful to have two sets of profiling experiments, one which is unreached in PTM and the other which only has the unenriched features. It is possible to also find some unmodified features after enrichment, but usually it's much worse quality. So having two separate runs is much more effective. So what we do in MS stats, we will have in TMT, we have one data structure for modified. We will usually have much fewer features. And then the data structure we talked about before for unmodified features, we will have the same model as before, separately for each data structure, no change. And now we will integrate and combine the output from statistical models so to adjust change in the PTM for the overall change in protein abundance. So This is how we do the adjustment. This is the fold change of PTM minus fold change of the protein. We can combine standard errors, and we can calculate the associated degrees of freedom, so the amount of information necessary for that. Let me just show you how it looks in practice. So here's an example from a human cell line, and it is an 11-plex TMT experiment. So the human cell line had uninfected cells measured at 0 and 6 hours, and infected cells infected at 1, 2, 4, and 6 hours. And so this is enriched ubiquitination, and also had separate global profiling runs. So this is one particular protein, and you see that, again, as before, the x-axis, these are different channels. The dashed line, vertical dashed line separate conditions, and I put blue boxes here around DOX 1 hour and DOX 4 hour. So the left-hand side, this is the ubiquitination, and so we can kind of see that maybe there is a difference, or maybe not, between 1 and 4 hours. So if we look at the 1 hour and 4 hours, you really can't tell. I would say probably there is no change in abundance. But at the same time, on the right-hand side, we look at the overall protein abundance, and we see that it decreases quite a bit between 1 hour and 4 hours. So if we don't adjust for protein abundance, it looks like there is no change in ubiquitination. But if we do adjust for change in protein abundance, it may actually be different. And so this is the result. So if we look at the volcano plot, this particular dot here is this protein we looked at just before, and it was not found differentially abundant, differentially ubiquitinated without the adjustment, but it is strongly significant if we account for the fact that the overall protein went down in abundance at the same time. And so here is the difference between adjusted and unadjusted changes in post-translational modifications. Sometimes there's no difference because the overall protein abundance didn't change, but there's also quite a bit of difference in some of the other conclusions. And the very last thing is the fact that we can actually use this approach with a graphical user interface. So MSStats Shiny allows us to access all of that functionality through the user interface. It's a standalone app, meaning that you can install it on your local computer. You don't necessarily have to use it on the cloud. We do have a server, but it has a fairly limited bandwidth, so it is actually best to install it locally. It can work with label-free and label-based experiments. It can do both global proteome profiling and post-translational modifications. So this is a list of different data acquisition types and a list of different data processing tools, with Proteome Discover prominently featured. So it is quite flexible in terms of what can be done. And this is the workflow. So you can essentially follow the same outline that I just mentioned. So you first select type of data acquisition, you select which type of file you will be reading after that, and so on. So it is very similar to what I discussed. Now, a very important thing, which I hope helps ensure reproducible research, is that as you select these options and you read the data in and you do the analysis, the app generates the R script that corresponds to the choices that you made, which essentially documents your workflow. And so what you can do later, you can take this R script and reanalyze the same data set and reproduce exactly the same results. So it ensures reproducibility, but it also helps one more thing. So let's say you have a really large data file, and it's really hard to explore it interactively. You can take a subset of proteins or subset of features and analyze it interactively like that, save the script, and then run the script overnight or on some bigger server to do the full analysis of the same data set. So this is very useful. So finally, we have a lot of publications just came out recently describing various aspects of MSStats. So we have the update to the MSStats 4.0. We have a manuscript describing MSStats Shiny. we have a general manuscript describing MStats TMT, and just recently the part about TMT experiments with repeated measures designs. This also came out in JPR just like a few weeks ago. And also we have a publication describing MStats PTM and also MStats LEAP. So there's quite a few resources that I hope will be useful. I, of course, need to acknowledge everybody who did the work, in particular Mina, Devan, and Ting, and Mateusz, who were really instrumental for MStats. And I would like to thank all the collaborators who provided data and feedback. We really value the support of our community and, of course, of the funding sources. And finally, the last disclaimer. And thank you very much for the attention.